In our last video, we talked about the different cells that float around in your blood vessels and organized them, and we also introduced some new cells that were part of the innate immune system. So now I want to review microbial evasion of phagocytosis. So we've talked about a lot of these before when we talked about microbial mechanisms of pathogenicity, but I'm going to review them now so we can sort of like come full circle and fit everything together. So right now we're talking about ways that we just talked about white blood cells. A lot of them are phagocytes. Some phagocytes are antigen-presenting cells as well. Now we're going to talk about how some pathogens evade phagocytosis. So M proteins is one. We talked about M proteins when we talked about, in Chapter 13, Microbial Mechanisms of Pathogenicity. So just to remind you, M proteins are proteins that you find on the surface of Streptococcus species. So this whole genus has this protein called M proteins. M proteins are considered a virulence factor. They prevent phagocytosis, okay? So they prevent pathogens from they prevent phagocytes from ingesting them. They're also, I don't have it on this list, but they're also adhesins. So they're a virulence factor not only because they help Streptococcus species evade phagocytosis, but also because they help Streptococcus adhere to host tissue. So those are two reasons it's a virulence factor. A third reason it's a virulence factor is because it destroys certain elements of the complement system, which is part of the immune system that we'll learn at the end of these lectures. So it disrupts the complement system, which is a component of the immune system. It's also an antigen. M proteins are antigens, meaning they can be bound to by immunoglobulins. Large capsules. That's another way microbes can evade phagocytosis. So if a microbe has a very large capsule, it can be hard for a phagocyte to physically reach its pseudopodia around the bacteria, especially given the negative charge repelling the phagocyte. So remember that some bacteria have capsules made out of glycocalyx. These are very large. It's difficult for phagocytes to reach their pseudopodia around. Also, that glycocalyx is negatively charged, and the cell membrane of all cells, white blood cells included, is negatively charged because of those phosphate heads. Like repels like. Remember, opposite charges attract, like charges repel. And so the glycocalyx physically repels the pseudopodia, the white blood cell and its pseudopodia, so that makes it harder to phagocytize as well. So when a bacteria has a capsule, a white blood cell, a phagocyte, will actually have to trap that bacteria against a hard surface so that it can't slip away in order to phagocytize it. It's not impossible to phagocytize it, it's just much harder. All right, so here's a slide about capsules. So some bacteria have capsules. This includes some Streptococcus species, Klebsiella. Those are just some examples, but a number of bacteria have these glycocalyx capsules. Bacteria with capsules are not ingested by phagocytosis unless in the presence of opsonins. So do you remember what opsonization is? It's something that antibodies can do. Remember, antibodies have 5 things they can do to help fight pathogens. There was agglutination, opsonization, and then a bunch, 3 others. So opsonization is facilitation of phagocytosis. That's when antibodies stick to a microbe, act like grips, so that it's easier for a phagocyte to grip the microbe with its pseudopodia, bring it into that bear hug to phagocytize it. Okay, so bacteria with capsules have to have opsonins like antibodies to phagocytize them. And the bacteria then get fixed against a hard surface like the alveoli in the lungs, some hard surface to allow the white blood cells to phagocytize them. All right, leukocidins. Some microbes produce leukocidins, which kill leukocytes. One example would be Staphylococcus aureus, which produces enzymes that break down the lysosome, spilling the digestive enzymes and acid from the lysosome into the leukocyte cytoplasm, okay, the white blood cell's cytoplasm. So Staphylococcus aureus, basically what that's saying is it can be ingested, but it can't be digested, so phagocytosis doesn't complete. So it's ingested into the phagosome, but then it excretes these enzymes that break the white blood cells, lysosomes, and then the lysosomes spill out their digestive contents into the white blood cell and kill the white blood cell. Okay, so lysosome destruction, that's one kind of leukocidin. So remember, anything with cidin at the end, like a pesticide, an herbicide, that's something that kills. A germicide. A leukocyte is something that kills a leukocyte, or a white blood cell. Okay. So just as our immune system can kill some pathogens by punching holes in them, some bacteria use that weapon as well. So some bacteria produce these toxins or enzymes that punch holes in a host cell, which ends up killing the host cell, so pore-forming toxins. So those are some examples of leukocidins that some pathogens have to prevent them from being phagocytized. And then prevention of phagosome-lysosome fusion. So some microbes, once they're inside the phagosomes, they can produce proteins that prevent fusion of the phagosome with the lysosome. So there's some examples listed here. These include pathogens which persist and replicate inside the white blood cells, inside the phagocyte. And they fill the phagocyte so much with their progeny that the phagocyte will then lyse and release a large number of microbes into the surrounding space. So HIV is a virus that does this. It can be it can be taken into a phagocyte through phagocytosis, but then once inside, it's not digested. Okay. Chlamydia is another example of this is a bacteria that can do this. Then you have Leishmania, which causes leishmaniasis, Plasmodium, which causes malaria. So this list has a virus example, bacteria example, protist examples. All right, so remember we are talking about our second line of defense here, just to get us back on track. So we've talked about the cellular defenders, and now we're going to talk about the point of inflammation and fever. So the whole point of fever and inflammation is to help phagocytes in their job of gobbling up pathogens.